May I present Biology Practical Paper 2005 KCSE. Welcome guys, welcome to my presentation. So welcome. All candidates and even those who are preparing for KCSE should not fail to see this video. So, Biology Practical Marking 2005 Paper 2. The time the paper was taking was 2.5 hours. And question 1 was as follows. You are provided with the specimens labeled S1, S2, Q, X, and Y. Details of what those specimens were is there in the confidential requirements video. The dichotomous key below can be used to identify the specimens. So we have the dichotomous key there. You know, dichotomous means two. Is a type of classification where you use two contrasting characteristics to identify a specimen. That's why it's dichotomous. All right. So you use it following those steps to identify those 1, 2, 3, 4, 5 specimens. And you can see now the way the dichotomous is. Key, leaves simple, you go to 2. Leaves compound, you call that one as Austrasea. Leaves green, go to 3. Leaves purple, Comerinaceae. Leaves parallel veined, Graminae. And leaves net veined, you go to 4. A, leaf margin smooth, you go to 5. Leaf margin serrated, you go to 6. 5A, leaves hairy, you identify that one as Solanaceae. Notice that these are Greek pronunciations, and of course we are trying our best. Leaves hairy, you go to 8. Leaves succulent, go to 7. While if leaves are not succulent, you have the identity Malvaceae. Seven A. Leaves with pointed tip, Classulaceae. B, leaves with rounded tip, Classulaceae. All right. Leaves ovate, Nyagatiginaceae. And leaves lanceolate, Anacandidiaceae. All right. The pronunciations are tough. But anyway, as long as you know how it works, you don't need to be scared by the words, because there's nobody who will tell you to memorize those words. They are always given in the examination, so your work will just be there to write what is given. So in biology, technical terms, we just test your knowledge on those technical terms. Using the dichotomous key, identify the specimen. In each case, show the sequence of steps, 1B, 2A, 3A, 6B, etc., in the key that you followed to arrive at the identity of the specimen. Here you get 10 marks. So, specimen S1 steps forward were 1A, 2A, 3B, 4B, 6B. You can go back to the key and see what the key is talking about there. And the specimen was Malvaceae. S2 was 1A, 2A, 3B, 4A, 5B, 8A. You identify it as Nyctaginaceae. All right. Q was 1A, 2B. Only 2 steps. And you get it. It was Comerinaceae. X was 1A, 2A, 3A, Graminaceae. You can call them Graminaceae or Comerinaceae, Malvaceae, all right, whatever, or CI or whatever. As long as you memorize what you can be able to spell. Astalaceae or Astalaceae. So Y is 1B, Astalaceae. Using the flowers, name the classes of the spermatophyta to which specimens S1 and Q belong. You get 2 marks. And S1 is Dicotyledonei. Reject wrong spelling, like writing dicot or dicotyledon or dicotyledonous, but proceed to mark the reasons. Q was monocotyledonei. Reject wrong spelling again, monocot, monocotyledon, monocotyledonous, but proceed to mark the reasons. Give reasons for your answer in B1 above. And the reason for S1, why it is dicotyledonei, is that the floral parts are in 5s. Or the floral parts are in, the letter 5, the numeral 5s. You can use the 1 or the numeral. Or the floral parts are in multiples of 5. Or you can say it has 5 stigmas. Or it has 5 sepals. Or it has 5 petals. Any of those was given a mark. Q was floral parts in 3s. So you can write the 1 to 3s, or you use the numeral and you write 3s. Or you say floral parts in multiples of 3. Or 3 petals, or 6 stamens. State how specimens S1 is adapted to its mode of pollination. Presence of large-colored bracts, conspicuous bracts, to attract insects. So guys, state how specimen S2 is adapted to its mode of pollination. Presence of large-colored bracts to attract insects. Presence of colored petals to attract insects. Scented to attract insects. Anther and stigmas enclosed in a tube to be reached by insects. Brightly colored bracts to attract insects. has current petals to attract insects, has current sepals to attract insects. All those answers have alternatives, and as usual, what I told you is that you cannot put an answer with a stroke. You choose one of the alternatives. Reject its brightly colored. All has bright colors. You are not getting anything from there. Give only one mark for have bracts to attract insects, since there is omission of brightly colored. Okay? Mark all the points because the stem of the question is open. So you mark all the responses, all the points, and then because the question was open, they had not told you state how specimen is adapted its mode of pollination and given you the number of points to give. So it is an open question. So open the flower of specimen S2, draw and label the pistil. The pistil is there. You can see it very clearly. All right. I can move the camera a bit nearer so that you can see. And you can see stigma, style, ovary, D2 and D1. Give one mark for the rebel pistil. D2, rebels divided by 2, 1.5, round off to 2. Magnification, so you are working length of drawing or image over length of actual specimen, all right, equals A units over B units, and the range is times 2 to times 10. You could give any of those. D1, continuous outline, no shading. D2, proportionality and accuracy. Longer, feathery or not, stigma. Thinner and shorter style than stigma and ovary, at least 1/3 to a half, oblong, long and wide ovary to get that D2. Note, flower stalk does not affect the marking. Reject any other drawing such as hypothetical drawings or whole flower. Accept demarcated diagram, accept flowing diagram. All right? So that is the way that diagram was supposed to be marked. Guys, you can see it even now, right? Question 2. Below are photographs labeled T1, T2 of specimens which were obtained from the same animal. Examine them. That is T1 and T2 are from the same animal. Okay. I'm trying to adjust this so that you see everything. With reasons, identify T1 and T2, 5 marks. Molar tooth. Reasons, presence of cusps or ridges, you can say in it, and then presence of 3 roots. T2 is incisor, chisel-shaped, wide sharp edge, wide wedge-shaped, wide cutting surface, wide cutting edge. All those were possible answers. In the photograph T1, label 4 parts of the specimen. So the 4 parts are very clear there. You can see cusp, or ridge, except cusps or ridges. Then we have the crown. There is a ridge of where you could have ribbon the crown. And then there is neck and the root. For crown and root, accept brackets if they do not overlap. No ribbon mark for any male because it is the outer cover of the whole tooth. There will be no mark for any male. Examine photograph body J with an inset of the front part of the lower jaw and photograph K with insets of front parts of upper and lower jaws. All right. I don't know whether it is visible to us, but I can take the whatever nearer. All right. Oh, excuse me, guys. So, guys, the camera fell down when I was trying to adjust so that you can see. I want you to see them properly. You can see photograph J. You can see the inset. All right. You can see that is J. 1, 2, 3, 4, 5, 6. The number of teeth are named there. Then you can see K. The same. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6. Guys, can you see that? Yeah. And the insets. Okay. So that is K. And that is J. Okay. So there they are. Giving reasons, state the diet of the animals whose skulls are shown in the photograph. J, vegetation, or grass, or shrub, or herbs, or plants, or leaves. Reject diet named herbivorous, but proceed to mark reasons. Reasons, presence of diastema, or that gap there. Absence of teeth, canines, and incisors at the front part of the upper jaw. Presence of horny band, or those. Presence of pronounced cusps or ridges for grading. Remember, take care of alternatives. Please give only one answer. Giving reasons, all right? Giving reasons. State the diet of the animal whose skulls are shown in the photographs. K, fresh or meat. Reject diet named carnivorous, but proceed to mark reasons. Reasons, presence of pronounced wrong curved sharp pointed canines for holding, for griping, for tearing, for grasping prey, any of those ones. Presence of carnaceous teeth for cutting and crushing bones, or you just say presence of carnaceous teeth. Label the canine tooth in the photograph, and there it is labeled, the canine tooth here. You can see it here, canine tooth. This one here. Any of those labels were correct. Or here, you could have named it here, accept any position for 2D. Okay, any of those positions, canine here or there, any of those positions you could have given, and you get 1 mark for canine tooth, 1 for pad. Pad, here, the label is here. You can see it here. The pad, this one here. can see my arrow there. All right. Okay. Write the dental formula of the animals whose scars are shown in the photograph J and K. The teeth that are not very distinct in the photograph are numbered, 2 marks. I showed you that the teeth were numbered. So you have I 0, 2, C 1, P M 33, M 33, I 33, C 11, PM 44, M 22, 42, and the other one 32. Note, letters identifying the teeth must be clearly written. Demarcation line of inoculum separating figures of the upper jaw and the lower jaw must be drawn, otherwise deny the mark, except if 2, 32, and 42 are not shown. That is multiplication of 2. You can see they are in brackets. So if the 2 and the other brackets there are not there, you can still give the mark. Identify the photograph of the skull from which the specimens labeled T1 and T2 could have been obtained. J. In appropriate diagram, label the position where the pad would be found in a living animal. 1 mark. It was shown in the diagram there. Accept a diagram drawn in the space or label the photograph 1M. So the pand was shown in the previous diagram and shown you. I think I showed you that diagram, this one. So the pand is here. Okay. Question 3. You are provided with the specimen labeled P. Examine the inner and outer leaves of the bulb. Reckon the differences between them. 1 mark. So inner succulent or juicy or fleshy, while outer is dry. Inner leaves thick, while outer are thin, papery, or membranous or scaly. Comparison must match to our mark, but I want reasons for mismatch. Fleshy and dry, succulent and dry, juicy and dry, thick and thin, thick and papery, thick and membranous, thick and scaly. A2 and A1 are tied. So if you fail A1, you cannot get A2. So A2. Inner swollen. Give reasons for the differences. Inner swollen with food for storage and outer for protection against desiccation or mechanical injury or excessive loss of water or microorganism or invasion by fungi. Remember, you give only one answer. You don't stroke. Reject both 1 and 2 if completely wrong. Question 3. Separate the roots and aerial leaves from the bulb. It's continuation of question 3. This is part B. So, crush the roots, aerial leaves, and the bulb separately. To each crushed material, add 1 ml of water. Using reagents provided, test for the food substances in each of the extract. Reckon the procedures, observations, and conclusions in the table below. So the table is there. Guys, I don't know whether it is clear, but I will try to read it for you. But it is clear because you can see extract, procedure, observation, and conclusion. Okay, the instruction is up there. All right. Using reagents provided, test for the food substances in each of these extracts. Click on the procedure, observation, and conclusions in the table below 9 marks. That statement is in the other place. So let's look at the table. Okay. The table, we have roots, bulb, and aerial leaves. In the first, for the roots, you have and 2 or any other measurement drops of iodine solution, no color change, starch absent. All right. To 1 mil or whatever, and equal amount of Benedict solution. We have blue to green to yellow to orange, so reducing sugar present. That is in the roots. You can see the other literature there. Bulb and the specified number you want of drops of iodine solution, no color change, starch absent. Aerial leaves, do the same, and drops of iodine solution, no color change, starch absent, but reducing sugar present, and also reducing sugar present in the bulb. So guys, you can see there. I don't know if I take my camera near, you can see, yeah, I think we can focus first on the roots. Roots, okay, look at the way it was done there. Rejecting, accepting. Then we focus the bulb. Okay, the bulb is there. Okay. And then we focus the aerial leaves. Guys, you can see the aerial leaves there. Fine, guys. All right. Note. But remember here, whatever I'm left with, you cannot talk. Okay. Note, if observation is green, conclusion should be traces of reducing sugar. Accept simple sugars for reducing sugar. Color changes to yellow without other steps of sequence. Reject conclusion if color sequence is wrong, e.g. green or orange to yellow. Observation and conclusion if procedure is wrong. Account for the results obtained in B above. Presence of reducing sugars translocated from the bulb or aerial leaves or fresh leaves for provision of energy or for respiration or for growth and development, metabolic activities, or for cell division or active transport. That's why they are there. Absence of starch because not a storage organ, because roots are not storage organs. You can also say that. Account for the results bulb, presence of reducing sugars translocated from the aerial leaves for storage to be stored. Absence of starch because fresh leaves of bulbs do not store starch. Aerial leaves, presence of reducing sugars due to photosynthesis. Absence of starch because reducing sugars had not been converted to starch. Part C must be marked in consultation with what is written in the table. The table must be correct to score marks in C. Goodbye, and see you next time for my next video on practical marking of 2006. Don't forget to look at the video on the requirements or the confidentials, which is coming out soon. Goodbye, guys, and good night.